Here is a very short lecture about the lymphatic organs. What are the lymphatic organs? They are the organs responsible for the immunity of the body. They contain a huge amount of lymphocytes. They are classified into two subtypes or two groups. Primary lymphatic organs, in which the immature lymphocytes get developed and matured. There are the bone marrow and the thymus. Bone marrow in which develop T and B lymphocytes. Thymus is for T lymphocytes only before puberty. After puberty, this gland becomes involuted and small and transforms into fibrous tissue. The secondary lymphatic organs include lymph node, spleen, and the tonsil. We are going to speak about the lymph node only. Spleen is not included in your study, in your and in your exam. The lymph node. What is the lymph node? As we know all, the lymph node is a very tiny organ, kidney-shaped or bean-shaped or maybe oval. The lymph node may be present singly, in single form, or in groups. These groups include the inside the neck, inside in the axilla, inguinal region, different areas of the body. Also, it is present intraperitoneal around the internal organs. As we know, any gland is formed of two parts. What are these parts? The stroma and the parenchyma. The stroma is a supporting tissue and the parenchyma is a functioning tissue. The stroma of the lymph node is divided into capsule, trabeculae, and the reticular framework. The capsule of the lymph node is thin, formed of connective tissue fibers and cells, surrounded by adipose connective tissue rich in fat cells, and some may have smooth muscles at the hilum. What is the hilum? The hilum as we see here, the lymph node is being shaped. The concave surface has a depression. It is the hilum. This hilum may contain some smooth muscle inside in the capsule of the hilum. The capsule also, to revise, it's formed of connective tissue fibers and cells. It is thin, may contain smooth muscles at the hilum and surrounded by adipose connective tissue. The trabeculae or the septa. The trabeculae is formed also of dense fibrous connective tissue, extends deep from the capsule, divides the cortex into regular compartments and the medulla into irregular compartments. They are formed of collagen fibers and fibroblast. The last part of the stroma is the reticular framework, which is formed of reticular connective tissue, which is it could be demonstrated seen only by using silver stain, cannot be seen using hematoxin and iodine. So, to summarize, the stroma is formed of capsule. The capsule is thin, surrounded by adipose tissue and may contain smooth muscles at the hilum. The stroma is formed of trabeculae connective tissue septa descending from the capsule, dividing the cortex into regular compartments and the medulla into irregular compartments, and the reticular connective tissue, which can be demonstrated only with silver. The parenchyma of the lymph node, as we mentioned before, is formed of cortex and the medulla. The cortex is formed of follicles, lymph follicles, sinuses, and paracortex. The medulla is formed of cords and sinuses. As we see here, the cortex is formed of follicles, sinuses, and paracortex. The follicles of the cortex or the cortical follicles are of two types. the primary follicles and the secondary follicles. What are the differences between the primary and the secondary follicles? First, the primary is the follicle which is not exposed to an immune stimulation. If this primary follicle is exposed to immune stimulation, it will turn into secondary follicle. What are the histological differences between these types? As we see here in this picture, the primary follicles appear homogeneous. What is the meaning of homogeneous? It is formed of uniform shaped small lymphocytes. The secondary lymphatic follicle have a germinal center with, as we can see here, paler center due to the presence of large lymphocytes. What are the types of cells present in both types? The primary follicle has densely packed small B lymphocytes without germinal center and macrophages. The secondary follicles have at the periphery dark, similar to the structure of the primary, plus the germinal center. What is the content of the germinal center? Large activated B lymphocytes, plasmablasts, plasma cells, and macrophages. So the difference is the development of large lymphocytes, plasmablasts, so plasma cells, which are not present in the periphery or in the primary follicle. So, as we mentioned, the cortex is formed of three components. The first is the lymph follicle. What are the rest? The second is the paracortex. Paracortex is the zone situated deep near the medulla. It contains mainly T lymphocytes and have that migrated from the thymus during maturation. This area have a post-capillary venules lined with simple cubical epithelium. As we know, the blood vessels are all lined by simple squamous epithelium. The only exception is the post-capillary venules, which are small veins, are lined with simple cubical epithelium. This lining had a very important role in immunity and development of the T lymphocytes, development and maturation of T lymphocytes. The cortical lymphatic sinuses, we have two types. We have under the capsule called capsular lymphatic sinus, and between the trabeculae are called trabecular sinus. They are lined with endothelial cells and macrophages. The medulla, the second component of the parenchyma, is the medulla. The medulla is formed of two parts, the medullary cords and the medullary sinuses. It appears paler in staining than the cortex as it is less packed with lymphocytes. As you see here, lymphocytes small amount and large amount of empty spaces, which are the medullary sinuses. It is formed of medullary cords that are irregular and branching cords of aggregated B lymphocytes, plasmablasts, and plasma cells. The medullary sinuses are the spaces between the medullary cords and trabeculae. They are lined by endothelium and macrophages. They contain lymph received from the cortical sinus. So how is the lymph enters and leaves the lymph node? As its function is filtration of the lymph. First, it enters through the convex surface by a lymphatic vessel called afferent lymphatic vessels. These afferent lymphatic vessels contain valves to prevent escape of the lymph when it enters the lymph node, and it forces it to pass in one direction only. Then it enters to the lymph node, it reaches the subcapsular sinus, then trabecular sinus to the medullary sinus, and filtered to be drained in the efferent lymphatic vessels that come out from the concave surface of the hilum. What are the functions of the lymph node? The lymph node functions include filtration of the lymph from microorganisms, proliferation and production of lymphocytes, and immunological functions. Thank you. I would like to remind you that spleen is not included in your exam. Thank you very much and best wishes.